Hello, welcome to Professor Monzon's World of Animals. Today we continue exploring the diversity of animal life. In this video, we will focus on the non-avian reptiles. Non-avian reptiles, what is that? Well, those are the reptiles that are not avian, reptiles that are not birds. What do you mean, Professor? All reptiles are not birds. All right, well, that's what we will discuss today and next time. So, uh, let's start by mentioning that we're going to follow the outline here from Hickman's Integrated Principles of Zoology, chapter 26. We'll start with a little brief discussion about enclosing the pond and what and kind of comparing the shelled egg of amniotes to an enclosed pond. Then we will talk about the origin and early evolution of the amniotes, focusing on some adaptations that amniotes have that amphibians don't, and a brief discussion about the changes in the traditional classification of reptiles. And then we're going to talk about two super cool groups of reptiles, the snakes and the maybe even cooler dinosaurs. Okay, what do I have here? All right, amphibians is the group that we discussed last. We know that amphibians evolved numerous adaptations to live on land, but with a shellless egg, thin, moist skin, gilled larvae, they remain dependent on water. This is something that we really emphasized last time when we were talking about amphibians. However, the evolution of a shelled egg freed animals to better exploit the terrestrial environment. So the evolution of terrestriality in the amphibian lineage was a tremendous step in the evolution of animals, of vertebrate animals. The evolution of the shelled egg was another one of these major innovations that permitted further exploration of land because it made animals less dependent on water. Extra embryonic membranes of the egg basically enclosed the aquatic environment to provide a complete support for embryonic development even away from water. So the pond, metaphorically, the pond that the amphibians were so dependent on, that environment was basically enclosed and maintained by a special membrane called the amnion, and that amnion protects the embryo in a fluid-filled sac, just like a private pond. These animals that have an amnion are called the amniotes. These are tetrapods that develop an amnion in early embryonic life, and these include the non-avian reptiles, the birds, and the mammals. So I wanted to talk about the origin of amniotes, and when we look at the fossil record, we observe an early diversification of amniotes that have three different patterns of holes in the skull. This is in the temporal region of the skull, so behind the eye socket. We have animals that have one fenestra. Okay, fenestra is the fancy scientific name for hole. So one fenestra in the head, and exemplified by this skull over here. So you see this is the eye socket, so that doesn't count as a fenestra. That's just an eye socket. But just posterior to it is this big old hole in the skull, and synapses have one per side, right? Obviously, there would be another hole on the other side. Um, An alternative is the diapsid skull, which has two fenestra. And so that is exemplified by this skull over here. You see the eye socket, and just posterior to the socket are these two holes in the skull. Animals that have two fenestra, modern animals that have two fenestra include the non-turtle reptiles and the birds. And then the other pattern that we see is the anapsid pattern, which means that there are no fenestra. So this skull, for example, has only an eye socket and no posterior holes. It is important to recognize that the earliest amniotes probably had an anapsid skull. So that was probably the ancestral pattern. However, the anapsid skull of modern day turtles likely represents a secondary loss of the temporal fenestra. So now we recognize two lineages that have these different patterns of fenestration. One we called Diapsida, and that is synonymous with Reptilia, and the other is Synapsida, and that is synonymous with Mammalia, the mammals. Both of those are monophyletic clades. Amniotes together form a monophyletic group that appeared and diversified in the late Paleozoic era. So here in this figure from your text, you see that here in the late Paleozoic, which actually extends further back than is shown here, but this is the late Paleozoic, we have a hypothetical ancestral anapsid that went on to diversify into various lineages. Okay, so this lineage over here, the synapsids, included many animals, some of which went extinct, and some others which did not and make up the modern day mammals. On the other hand, there were some other animals with two holes in the head, the diapsids, and that lineage diversified greatly. Some of those branches went extinct, and some others persist in modern day reptiles and birds and crocodilians. So now we recognize three branches of diapsids that persist to this day. One is the archosaurs, right? So it's these animals. Some of them went extinct, but some did not. And the ones that did not are the birds and the crocodiles. Another group of diapsids is the lepidosaurs, and those include the modern day lizards and snakes and totaras. And then the other group is this weird group of animals called the turtles. One of the take-home messages of this figure is that the diapsids diversified greatly in the Mesozoic era. Okay, this is the era that we also call the Age of Reptiles. It was really long; it lasted 165 million years, and in that period, the reptiles really flourished. And they occupied the land, but also the sea. Check out that there was this other lineage called the plesiosaurs, which were aquatic reptiles, sort of like the Loch Ness monster, and they went extinct. And then there are these other bizarre dolphin-looking reptiles called the ichthyosaurs, and they also went extinct. Many lineages went extinct at the end Cretaceous mass extinction about 66 million years ago. And so you see here in this boundary, the plesiosaurs went extinct, the pterosaurs went extinct, and many dinosaurs went extinct. When we look at this cladogram, there are a few take-home messages here. One is, well, what I said earlier, that there are these three different patterns of fenestration. The amniotes that have a synapsid skull went on to become the modern day mammals. The amniotes that went on to have a diapsid skull went on to become the modern day lizards and birds. Notice that the turtles are diapsids phylogenetically. They're diapsids. However, morphologically, they experienced a loss of diapsid openings in the skull. So they have an anapsid skull. However, they're still considered diapsids. Sort of like snakes are considered tetrapods, even though they don't have limbs. Another of the take-home messages from here is some of these basal characteristics that unite the amniotes as a monophyletic group. And here they are, those extra embryonic membranes of the amnion and the chorion and the allantois, which we'll talk about shortly. That's basically the shelled egg. And notice here that the group of non-avian reptiles that is most closely related to the birds is the crocodilians. Okay, which leads me to the next question here. I'm going to disappear because I want to give you some time to think about this. Are birds reptiles? Okay, I'm gonna come back. I'm not sure if the timer went all the way, but I wanted to give you some time to really think about this, to kind of ingest and digest this cladogram. All right, well, to answer the question, are birds reptiles, we have to, well, first define reptiles. It's very similar to the question, are humans apes? Okay, well, then how do you define ape? Remember that there's um, the traditional classification that is more emphasizes more body form, and then there's the more modern cladistic classification that emphasizes evolutionary relationships. So traditionally, since the days of Linnaeus, reptiles were these like scaly animals, right? The snakes, the lizards, the turtles, and the crocodilians. But now the morphological and genomic evidence shows us that the crocodilians are actually more closely related to the birds than they are to other animals traditionally called reptiles. So that means that if we insist on a cladistic approach, we have to actually redefine reptiles. Because if we exclude the birds, that would make reptilia a paraphyletic group. So the easiest thing was to redefine what we mean by reptile by including the birds. Okay, that way it's a monophyletic group. Here they call it diapsida. Diapsida would include all the extinct forms of diapsids, uh, but remember diapsida is synonymous with modern day reptilia. Okay, so talking about the amniotic egg, with its extra embryonic membranes. Okay, the extra embryonic membranes are the amnion, the allantois, the chorion, and the yolk sac. The amnion, as I mentioned before, is that membrane that encloses the embryo in fluid, providing that aqueous medium for growth. It is where the developing embryo is. The allantois is a sac that stores metabolic wastes in the egg, right? As this little animal is growing, it is generating wastes that needs to be excreted. Well, they are stored in a separate sac, the allantois. The chorion is an outer membrane that surrounds the contents of the egg, and then the yolk sac contains the yolk that provides the nutrition. Both the allantois and the chorion function in respiratory gas exchange. Okay, so while all the food of that embryo is going to need is in the yolk sac, all the oxygen that embryo is going to need is not in the egg. It is being exchanged with the external environment through the allantois and the chorion. And of course, there's the shell, which provides structural support in a semi-permeable membrane that permits the passage of gas, but not of water. To develop this shelled egg, animals require internal fertilization and thus a penis in many lineages. And I want you to contrast this with the external fertilization, which is the typical form of fertilization in the amphibians. I've mentioned several times that mammals are considered amniotes. Well, where is our shelled egg? Well, most mammals have a placenta, and the placenta itself is a modified egg. The embryo is still surrounded by an amnion filled with amniotic fluid, just like in a regular egg. But the allantois and the yolk sac become the umbilical cord, providing the connection through which food reaches the fetus and wastes are removed. So together with part of the chorion, all of these membranes make up the placenta. Let me ask you, what breaks when a woman goes into labor? When a woman says that her water broke, what exactly is it that breaks? Another adaptation of amniotes is that they have thicker and more waterproof skin. And amniotes possess numerous body structures that are composed of keratin. Keratin is a uniquely vertebrate protein. And many different things are made up of keratin, such as scales and hair and feathers and claws that project from the skin. So reptilian scales project from the skin, and check this out, they are not homologous with the bony scales of fish. Some reptiles, like crocodilians, for example, they do possess some bony plates that are called osteoderms, but those are different from the typical reptilian scales. That the reptilian scales are keratinized. As you see in this figure, here in the outermost layer of the skin, the epidermis, you see the scale that is made out of keratin, and underneath is in the dermis, which is a deeper layer of the skin, you see the bony osteoderm. So these are scales made out of two different materials. Keratin is really, really tough. So it protects the skin from physical trauma, and it's also covered in hydrophobic lipids that prevent the water loss through the skin. So these changes in the skin morphology made the skin of reptiles tougher, but because it's tougher and thicker, it prevents the skin from functioning in a respiratory gas exchange like we saw in the amphibians. Right? So amphibians have much thinner skin through which oxygen and carbon dioxide can pass through, but because it's so thin, it doesn't offer them much uh, physical protection. Okay, another adaptation of the amniotes is their efficient respiratory system. The lungs of amniotes are much more developed and have much more surface area than amphibian lungs. When we dissect the frog, we'll see that the lungs are tiny, but the lungs of amniotes are really large. Think about our own lungs, or the lung in this crocodilian in this diagram. It's a fairly large organ here occupying this thoracic cavity. And it's not just the size of the organ, but just how the organ fills with air is different. It's more efficient. Amniotes draw air into their lungs by aspiration, right? By sucking and creating this negative pressure by expanding the thoracic cavity using the muscles of the costal muscles. Those are the muscles in between the ribs, and also the diaphragm. It creates this space that allows air just to flow in, and that's different from the way that amphibians fill their lungs. Amniotes also have stronger jaws. Think about the jaws a a lizard about this big compared to the jaws of an amphibian of about the same size. The lizard is going to have much stronger jaws. Part of that is because of the way that that reptiles feed, but morphologically it's part of the way that the skull is constructed. Remember those temporal fenestra, those holes in the head? Uh, those holes are there for a reason. They basically permit the expansion of the musculature of the jaw, resulting in a mechanical advantage and a much greater bite force. So, for example, in this figure, you see the kinetic skull of a monitor lizard. And so, normally, a jaw is hinged only here so that the jaw, well, let me show you, let me open my camera, so that the jaw can just open like this, right? But in many diapsids, the jaw can open like this and then like that, and then it can close shut, creating a much stronger bite force. So basically, diapsids have more reticulations in their jaw um, that allow them to have a wide gape and a strong bite. Another adaptation of the amniotes is that they have an efficient cardiovascular system. Basically, all amniotes have completely partitioned atria and completely or almost completely partitioned ventricles. Remember, fishes only have one atrium and one ventricle, and amphibians have how many? They have two atria and one ventricle. Well, amniotes have two atria and two ventricles. So this permits the separation of circuits, prevents the mixing of oxygenated and deoxygenated blood, keeps blood that is rich with oxygen separate from blood that is low in oxygen, and makes for a more efficient circulatory system. What it basically does is it allows the animals to be more active and have a higher metabolic demands. Well, with more metabolic demands also come more waste. So they had to adopt a more efficient excretory system. So amphibians excrete ammonia. Ammonia is toxic and must be diluted, but this is not adaptive for living in dry terrestrial habitats where water is more limited. So reptiles, including birds, excrete these nitrogenous wastes in a different form, in in the form of uric acid, which is non-toxic and requires little water for excretion. So we are all familiar with the uric acid that is excreted by reptiles from the bird lineage of reptiles, and some of us have had cars that unfortunately we parked in the wrong place at the wrong time, and they wind up looking like this, full of, where the roof of the car is full of uric acid. And the last thing that I want to discuss as an adaptation of the amniotes is their expanded brain and sensory organs. Amniotes, and especially mammals, have pretty large brains. They have a large cerebrum and a large cerebellum. This allows for a better integration of sensory information and a better coordination of muscles for locomotion. So amniotes um, uh, have very well developed senses, and they can also very efficient in the ways that they can control their bodies and coordinate their muscular movements. And they can also, some of them have evolved intelligence. Okay, now I want to talk about two really cool groups of reptiles, of non-avian reptiles. First, let's talk about snakes. How do snakes sense their environment and locate prey? Well, let's talk about the sensory powers of snakes. Well, snakes, for the most part, have pretty poor vision, except for arboreal snakes that have really good binocular vision, so they can locate and chase prey through this highly three-dimensional complex environment, which is in the branches of trees. Most snakes have poor vision, and they have poor control over their eyes, which is why it seems like they're always staring, which is what one of the reasons why people get the creeps with snakes is because it looks like they're always staring. Snakes also don't have very good hearing. They don't even have ears, so they don't have external ears. They do have internal ears, but their hearing is very limited to a fairly narrow range of frequencies, from about 100 to 700 hertz. That's really narrow. To give you context, the human ear is sensitive to a much broader range of frequencies, from about 20 hertz to about 20,000 hertz. Now, the average human voice is about 250 hertz. So if you ever see a snake, don't talk bad about it because it can't hear you. Now, uh, even though they can't see very well and they can't hear very well, their ventral side is very sensitive to vibrations on the ground. So they can sense very, very small vibrations of, like, for example, an animal moving along the ground several meters away. They do have a very well developed Jacobson's organ, and this organ is in the roof of the mouth, right? And that little organ is lined with richly innervated chemosensory epithelium. Ugh, what does that mean? Okay, that means that it can sense chemicals. Basically, they can taste chemicals. And that is why this copperhead over here is flicking its tongue. It's basically flicking its tongue to pick up chemicals in the air, and then it's going to bring its tongue into its mouth and brush it against its Jacobson's organ, which will interpret those chemicals. So it's essentially like tasting chemicals in the air. Another organ that is very well developed in many snakes is the pit organ that is between the nostrils and the eyes. So in this rattlesnake here, you see here the eyes, here are the nostrils, and then here is this pit organ. It's also present here in the copperhead right over here. And the pit organ is lined with infrared sensing receptors that allow vipers to see heat. It's a really cool sense. It's almost like the Predator. Have you seen that movie? You should definitely see it. Don't watch Predator 2, but definitely watch Predator 1 for homework. Uh, the pit vipers have a pit organs that permit them essentially to see heat. Okay, how do snakes catch and kill prey without limbs? Imagine you trying to go out and catch something to eat, but you can't run, and you can't, and once you and you can only just bite it. And once you bite it, you can just try. You need to try to manipulate your mouth around this thing that you just caught. It's really difficult. Well, snakes have three strategies. They can capture and swallow live prey. This usually works okay with small prey, but for larger prey that may have some sort of defense, or maybe like scratching or biting to try to defend themselves, it's better to kill them before eating them. And so some snakes resort to constricting their prey, like this poor mouse is being constricted by the snake, and some other snakes resort to injecting venom into their prey and then waiting for them to die and then eating them. Now, the saliva of the saliva of non-venomous snakes possesses limited toxicity, and it is believed the that is the ancestral state of snakes, because we know that the venom gland of venomous snakes is a modified salivary gland. So in this figure, for example, you see here the fangs that are modified teeth that are hollow, and they are connected via this duct to these venom glands. Okay, these venom glands are modified salivary glands, and there are basically two types of snake venom: neurotoxins, which we've already talked about. Those are substances that attack the nervous system, essentially paralyzing their prey. And then there are hemotoxins, which basically attack the circulatory system and basically cause massive hemorrhaging. Now, venomous species occur in various snake families. For example, the family Viperidae, which are the vipers, like the rattlesnakes, the copperheads. They're all vipers and very venomous. The elapids, very venomous family of snakes, including the mambas and the cobras. And then there's a huge family of snakes, the colubrids, which most of them are non-venomous. However, there are a few that are venomous, such as the boomslang. Also, let me ask you this: so, if there are s- species that are species of snakes that are venomous distributed in various different families, is this a case of parallel evolution or a case of common ancestry? Homology. I'll leave you with that question to find out on your own. So, how do snakes swallow prey much larger in diameter? Okay, remember I talked about the articulated skull of diapsids? Well, snakes take this to a greater degree because the bones of the skull are very loosely articulated in snakes, so that the entire skull can flex to accommodate oversized prey, as you can see here in this egg-eating snake that is eating an egg. Basically, the two halves of the lower jaw are joined only by muscle and skin. So while our jaw is just this one continuous structure, in snakes, basically their chin is very soft, right? So you have the left dentary and the right dentary are only connected by flexible muscle and skin. So it allows them to manipulate their jaw, each half of the jaw, independently. They also have these posteriorly pointing teeth that prevent prey from escaping as the snake slowly advances its mouth over the prey. So the snake, once it catches something, that thing usually cannot escape, right? And so, for example, this is a python, so this is not a venomous snake, right? It just needs to hold on to its prey that may be struggling, and the more that the prey fights, the more that the snake's teeth get into the prey. And essentially, the snake will work half like the left side of its mouth, then the right side of its mouth, and the left side of its mouth, and the right side of its mouth. So it's essentially almost walking its jaw around its prey. All right, I want to show you a video here from this is a video of a Gaboon viper, which is the species of snake that has the longest fangs of all snakes, about 2 inches long. Viewer discretion advised.
Any number of potential prey could venture past. Velvet isn't picky. She'll eat anything from rodents and birds to small antelope and monkeys. A slight miscalculation makes no difference. Once she's set her sights on a target, there's no chance for escape. Her strike isn't the fastest in the snake world, but at over 20 feet per second, it does the job. The shock of the attack stuns the bird, while the complex venom cocktail rapidly floods its body. As she waits for her prey to die, Velvet adjusts her fangs. They can really get in the way sometimes. Within 10 minutes, it's all over, and lunch is ready. Eating is a specialized process. Velvet unhinges her jaw to allow her to open her mouth wide enough. Using rows of teeth to latch on, she drags her prey to a safe place where she can eat in peace.
All right. Did you like Velvet? Pretty cool name for one of the most dangerous animals in the world, one of the most dangerous snakes in the world. So Velvet is a Gaboon viper, right? So she belongs to the family Viperidae, which has those pit organs. So Velvet would have been able to locate and strike that her prey in complete darkness because again, she's not relying on vision. She's relying on those on that heat sensing um, ability where she would have been able to see the heat from coming from that bird even in complete darkness. It's a really cool creature power. All right, let's end our discussion of the reptiles by talking about one of the coolest group of reptiles that has fascinated scientists and non-scientists alike, the dinosaurs. Uh, it was Richard Owen, the famous English anatomist, who coined the term dinosaur in 1842. Okay, what else was happening in England in 1842? There was this guy running around, Charlie Bob. Remember, he had just come back from this voyage around the world on the Beagle, and he was working up this theory. Now, Richard and Charles knew each other. In fact, they worked together. In fact, it was Richard who analyzed some of the fossils that Charles obtained in the Americas. So they were sort of collaborators. Richard Owen did not agree with Darwin, not on religious grounds. Well, partly on religious grounds, but partly on scientific grounds. He thought that evolution, if it did occur, must have been different than by natural selection. So they were sort of rivals. Anyway, Richard Owen was a very good naturalist, very good paleontologist, and he created the term dinosaur. And now we know that there are basically two lineages of dinosaurs. There are the uh, ornithischians and the saurischia. Okay, what does that mean? The the ornithischian dinosaurs. Well, the word means, ornith means bird, and ischia refers to a bone of the hip called the ischium. So it means bird-hipped, and those are these dinosaurs right here. So these dinosaurs have a particular kind of a hip architecture that is similar to modern day birds. And so that's why these are called the ornithischian dinosaurs. And the others are called the saurischia dinosaurs. And they have a particular type of a hip um, that is similar to modern day lizards. So they are lizard hipped. Now, traditionally within the Saurischia group, there are two groups that are recognized. They are the sauropods. Those are the humongous herbivorous and quadrupedal dinosaurs like here Titanosaurus and Brachiosaurus and Argentinosaurus. Um, Those are the really really large ones, um, that with very very long necks and tails, and probably moved very slowly. The other group of saurischians are the theropods, and the theropods were all carnivorous and they were all bipedal. For example, like the velociraptor, like the much larger and muscular Allosaurus, and like the much larger and, well, a little bit larger and much more popular Tyrannosaurus. So I do want you to know that within dinosaurs there were uh, those several lineages. Now, as I mentioned before, dinosaurs and really diapsids in general, they flourished during the Mesozoic. That's why the Mesozoic was called the Age of Reptiles. But they went extinct at the end Cretaceous mass extinction 66 million years ago. Or did they? I'll leave you with that cliffhanger that we will discuss next time. Until then, be strong and courageous.